Okay. So everything fine? So you can hear me? Good, perfect. So, okay, welcome to my second lecture. And we are already well prepared from yesterday's talk. So because all the methodology we need for today, I have introduced already yesterday. So today I would like to speak about really our current research. And this is rate-dependent critical phenomena in ecosystems. And so you see a lot of co-workers, this time also some Indian co-workers, as you see. So I have a long-term collaboration with Shamal Dana and his students. And let me give you a motivation why we are interested in this. So because this is a workshop about climate, and so there are several threats to ecosystems in the course of climate change. And I would like to emphasize two of them, which I would like to address also in the talk. One of them is loss of biodiversity. So there are lots of species which are getting lost. And when we see here, so to say, on this view graph, so this is the time, so to say, these are different cultural time zones here, so which are very, so to say, going very much to the past. So here is, for instance, gatherers and hunters, then agriculture period, and then this is the time now. So this is the human population. So human population grows like crazy, as we see. But at the same time, these are the same periods here. We see that biodiversity declines quite a lot. So these are marine mammals and pelagic fish. I have only taken from this paper the ones so which are related to marine biodiversity, because I'm in the Institute for Chemistry and Biology of the Marine Environment. So therefore, we are mostly interested in this kind of species, but this would apply also to other species as well. Now, so there is already a tendency, so to say, recently I have seen papers which already talk about the sixth period of mass extinction. So there have been mass extinction periods before, so in the very past, but now, so to say, people already think about that we are in a phase of another mass extinction. Now, the possible causes are basically known. So there are lots of different threats here. So eutrophication is one of them. Most of this is man-made, we have to say. And so man-made on one side, but also climate change, which is also man-made, does its share. So eutrophication, that means we put more and more nutrients into the rivers, into the sea, so which change, so to say, the ecosystems. Then we have bioinvasion, so there are species which are moving when you have climate change. So a species has basically three possibilities how to react on climate change. Either you adapt, or you move into another region where the climate is better for you, or you die. This is all what you, what you can, so to say, expect. Then we have a lot of exploitation, of course, of our marine resources. We all like eating fish. And so this, of course, also is a problem. And then we are changing habitats all the time because urbanization comes along with changing, so to say, land use on a global scale. So this is one of the threats. Another threat is shown on this slide. And this is the increase of harmful algal blooms. So these are species which are present in the ocean already for a very long time, but usually they are present in very low concentrations. So therefore, they don't disturb much. Why they are harmful? So they are harmful for different reasons. So some of the species are harmful because when they start blooming, so they are in such a huge abundance so that they consume all the oxygen, for instance, or so to say, they are consuming all the nutrients. So that means they are changing basically the whole ecosystem dynamics. Others are really producing toxins, which are toxic to other species. So it goes up, so the toxins travel up the food chain, up to the fishes, and then you see mass extinction of fishes in certain areas due to this harmful algal blooms, and it can also go into our body, so this is also a threat for us because it ends up on our plates. And here you see some examples. So it's all over the world: New Zealand, Baltic Sea, US, California. So these are different species here. And you see, so to say, they change the color of the ocean. So this is just to make you make clear, so to say, in what abundances they really come in such a bloom. So they are so abundant that they really change ocean color. So this is just to get an idea. But they were in the past very rare. So this is a time course here over 10 years in the North Sea, and there is 1 in 10 years. Very strong bloom. So there is a normal spring bloom, which usually happens. This is the toxic bloom. Here it's 1 in 10 years. So when we look at this, these red dots are harmful algal blooms in 1970. So you see a little bit of scattered dots here. This is the same picture, 2009. So we see it's all over the world. It has spread all over the world, and the blooms become much more severe. So this is a real threat which comes along with climate change. So, and therefore, so we have to think about whether this theory of critical transitions can contribute to an understanding why we have, or what are the mechanisms of these different threats. Yes, please. I could not understand you because you don't have a microphone. Can you say it again?
The diversity is quantified, and there are measure or
they are just counted. Yeah, they are just counted. So it's a real tedious work. So I have visited a laboratory and tried to count myself algae species, for instance. It's a real tedious work, and this is done really manually. So you look. So there is, of course, also nowadays for plankton species there is some automatic counting also. But so if you really want to go for all the species so that you can differentiate that, you have to count it manually. So they are just putting this in the microscope and look through the microscope and count. It's a very tedious work. So when you see the data, then, and you did it once yourself, then you get a great respect for every data point in this time series. So, okay. So we are back to where we have been yesterday. And so what I would like to show you is that, of course, we want to understand what are the causes or what are the possible mechanisms for loss of biodiversity, for instance, harmful algal blooms, that you have a breakout of populations. So it's not only harmful algal blooms, breakout of certain beetles which destroy forests, which is a very threatening thing also in Europe. So it's the same category of idea here. And this is what we would like to study. So can we use ideas from critical transitions to explain these two phenomena, and how does it work? So when you look into the literature of critical transitions in ecosystems particularly, then most of the papers are devoted to multi-stable systems, so that you have different stable states. And as I already mentioned yesterday, so ecologists are debating very controversially whether this is really true, because the data do not show very much of this multi-stability, and therefore there is a big debate whether this theory can contribute anything. So therefore, we are concentrating on these systems which have one stable state, but of course also two stable states we have in mind. And as I mentioned yesterday, we have to take climate change as something which happens on a rather fast time scale, and this time scale is comparable with the time scale of ecosystems. And therefore, we need to look at non-autonomous systems to deal with this. So, okay, I explained already yesterday what a rate-induced transition is. Here you see another cartoon picture, so to say. So, because it's a critical transition, and I have taken as a cartoon only the one where we have basically a critical transition with one stable state. So there's no bistability here, but you can think of it that, so what we yesterday had is that we distinguished two types of behaviors when we change parameters or forcing in an ecosystem. So there was one trajectory which could track the stable state. So, so this was one of the opportunities. And the other one was that it tips and goes into another area. And in this case, it is another area in state space with different properties. So this is why we have a tipping. And this is what we saw yesterday. And this is now shown in this cartoon again. So where we have an environmental change, so to say, in time with a fast trend. And we visualize this fast trend now by taking our stability landscape. And so the stability landscape itself doesn't change, so to say, but we move it as a whole. We move it as a whole. And then you can think of a ball which lies in this equilibrium. And when we move the landscape with a certain speed, and so then, of course, this ball has a kind of inertia, and it will, of course, not follow immediately, so to say, the move of the landscape. So it will go to some other location here. But when it's not too far away, so then it would go back. And this would be a tracking phenomenon. But if we do it very fast, so then it could be that the ball is going to some completely different region in state space and is taking a very different trajectory here, visiting parts of the state space which have completely different properties. And then this can give rise to tipping, as we discussed yesterday. Okay, so these kind of excursions we are interested in. And so let me start so with an example. So where we look at these harmful algae blooms, this was the second example, the second threat, which I already mentioned. And so we would like to study this, whether we can see that this type of harmful algae blooms can be explained in terms of rate-induced tipping. So can we induce by changing environmental conditions very fast, can we change the dynamics in such a way that we have such a very fast blooming of a certain species? Okay, so we look at the model which looks very similar to models which you might know from a different context. This is from neuroscience, because this is an excitable system. So you can model harmful algae blooms with a model which resembles the dynamics of a neuron, because it has a very fast rising bloom of phytoplankton here. So this is an interplay between phytoplankton and zooplankton. Phytoplankton, these are the green algae, so which you have all these harmful ones, the red ones we saw here in the previous photographs. So they rise very fast. Then the grazers, which are suppressing these species because they eat them, so they slowly grow, and at some point, the grazers have such a concentration that they just eat up the bloom. And so this happens under certain conditions, and this is a system which is controlled by the zooplankton because it's a kind of top-down control. The grazer is basically responsible for eating up the bloom. So the night lines are shown here, so they look quite similar to what a Fitzhugh-Nagumo model would look like, so which is probably familiar to some of you. So we have a cubic function here, and then we have one linear function, and then there is only one fixed point here. So this is the stable fixed point, and there is a coexistence of predator and prey in this case, and this means phytoplankton and zooplankton. And it depends on the initial condition, so whether you get a bloom, because you see the zooplankton has to be low in concentration, because then there is no grazing pressure, so then a bloom is initiated. If there is too much zooplankton already in the system, then they can immediately suppress this bloom by just eating it up when it starts growing, so therefore it doesn't grow at all. Okay, so now what we have learned yesterday, so we would like to apply now. So let's change. So this is our model system now. And so it looks quite different, as you see, from the Fitzhugh-Nagumo equations. So this is really appropriate to population dynamics, but the null-lines look almost the same. But we have now our two species interacting, but we are changing now the carrying capacity. So that means the carrying capacity is more or less the environmental conditions for the phytoplankton, so for the prey, and we change it. We change not the carrying capacity we could do, but I just saw that I have chosen the case where we change the growth rate, because it's a lot faster than the carrying capacity, so you can exchange both of them. But so in terms of climate change, the growth rate is more important, because global warming will change the growth rate, and it will increase. And so therefore, what we have done is basically we have mimicked, in a way, a change in global temperature, and then there is a rise of the growth rate, and then it's constant over time. So because it cannot rise, so to say, forever. And then we extend our model system by one equation, so which mimics the change of the growth rate. So what happens? So what happens is that there are different behaviors or different, say, responses of the system to global warming. So because this is a kind of global warming what we have here. And then we see if this goes slowly. So species can adapt, basically, and they follow the steady state, which would be the gray one here. They follow it somehow in the neighborhood. So that means this is a kind of tracking. And if it's too fast, so now our speed is much faster, then we see that a bloom occurs. Indeed, so to say, we see that rate-induced tipping yesterday we had a population collapse, but here we have an outbreak. We have an outbreak of a population, and rate-induced tipping can initiate such a bloom and could cause a bloom. If we have a heat wave in the ocean, and they are also becoming more and more frequent, then this is what we could expect if such species are there, which have a very large growth rate, so to say, having this kind of blooming behavior. And so this would be the same thing now in dimensionless quantities, so it doesn't change much. So this is the critical manifold. We had yesterday discussed the critical manifold theory. So now we see we have again the same type of picture as we had yesterday. So we have this folded critical manifold with the stable parts here in red again, the unstable parts in blue again, and our trajectory, this would be a tracking trajectory here, would stay close to the steady state, which is very close. This is this dashed line. It's very close to the other fold, because now we have two folds, as you see. We have one here, and we have one here. So there are two folds in this system, and but for the blooming behavior, only the one here is the responsible one, as you will see in a moment. So now this critical manifold, as we discussed, here is once again a closer look up. So we are here now with an initial condition, because what we learned yesterday is that it depends on the initial condition and on the rate. So whether the trajectory will track or it will tip. And since this is a tracking trajectory, we see so that this one here is staying in the neighborhood. This fold here is repelling. So that means even if the trajectory, so to say, comes close, it is repelled. And basically, the trajectory stays in the neighborhood of its original steady state. That means this is a tracking trajectory. But if I increase now the rate, so then the trajectory will, so to say, tunnel under this, if you like. So we tunnel under this fold to the other side of the critical manifold, because we go then now from this part of the stable manifold to this part of the stable critical manifold. And the trajectory is just, so if this would be, so to say, the dip, so the fold which is here, the trajectory just goes under and slips to the other side. And then it makes a huge bloom, comes back here because it will also, so to say, cross once again this fold and go back to the critical manifold here and goes back to the steady state. So that means it develops a bloom if the rate is large enough. So here we see the picture once again, because what we learned yesterday and what we see here again is that so it depends on the initial condition, and we have learned that there is a special trajectory, the Carnot trajectory, which separates the tracking and the tipping initial conditions. So, and this is what we see here as well. So there is again a Carnot trajectory, and what we didn't discuss yesterday is that this Carnot trajectory, which separates the tracking from the tipping would be here, but it's not shown. So, so this Carnot trajectory is also moving when I change the speed of my environmental change. So that means a trajectory which starts in an initial condition, which is tracking, and I increase the rate, it turns from a tracking trajectory to a tipping trajectory. So it's the same initial condition here. So now the rate is faster. So that means, so this thing here has moved. So this trajectory, so this is not even seen this point anymore because it's somewhere here. And so that means this initial condition here is now in the area where it tips. And that means even with the same initial condition, with a higher rate, I would go to the tipping phenomenon, and then I would tunnel, so to say, under this fold here of the critical manifold to the other side to develop a bloom. So the mechanism we have already discussed yesterday. So what we have to do to get the critical speed, because the critical speed is, of course, exactly at that point, so to say, when this blue curve, the Carnot, is exactly on this initial condition. So when then the Carnot is passing by, so to say, is passing this initial condition, when it's directly on this initial condition, then we get the critical speed. So there is a critical value, again, like in a normal bifurcation, but it's a critical value for the rate and not for a value of a parameter. This is the crucial difference. And it is also clear that for each initial condition, the critical speed is different. It's not the same. So because we see that this guy is moving, but it's not something which is a simple curve. So this can be a complicated curve, and that means my initial conditions have each of them a different critical speed. And then we end up, so to say, computing this critical speed by computing the Carnot and computing when the Carnot is basically meeting our initial condition, then we get the critical speed or the critical rate. So here's also an illustration once again of what we saw yesterday already, because these critical manifolds are usually, because it's much easier, computed when the timescale separation is ideal. So that means we have an infinite time timescale separation. So our parameter, which describes the timescale separation, goes to 0. And then these manifolds are here quite nicely. So you see here our canal. Then we have the part where it's repelling. We have the part where it's attracting. But then, so to say, the trajectory goes through here. And when we have the normal timescale separation, so it's not the perfect one, so then we still have a canal here. But we see now what we had already in the illustration yesterday, that these critical manifolds are not matching, again, this fold here. So they are tilted now. And this tilt is only, so to say, they are meeting only in the point so where the canal is. So the canal is really an exceptional trajectory, so which determines basically the dynamics here. Now, so now let me get to the formation of multiple plankton blooms. So because this is what we saw. So there is one thing, so what we can explain, okay, rate-induced transitions are able to form a plankton bloom. So a harmful algae bloom can be explained by global warming so that we have such a bloom. And now we have also seen in one of the first slides I show that it's not only that we have harmful algae blooms, because we had them in the past also, but very rarely, but we see also a lot of them. And now we can also explain that. So when we increase the rate, so we make our climate change faster and faster, and this is unfortunately what we are doing, just by man-made changes to the climate, then what happens is then if we have a bloom and it's fast enough, so our environmental change is fast enough. The environmental change is basically here. This is this axis. So we are moving along this axis very fast. And then the other variables in our three-dimensional state space now are the following. So first, we develop a bloom, but then we end up in a location on the stable critical manifold, which is still before the canal. So that means there is, so I'm still before the canal, and that means that further environmental change, because we are changing, so to say, with a quite high speed, would lead to the next bloom. And it would stop only, so to say, when we are beyond the canal, so where we go to the area, so where we have tracking behavior. So this is also a mechanism so that we can form multiple plankton blooms. It only depends on the rate of climate change. So when we change the temperature too much, then more blooms become possible, and this is what we see at the moment, so in observations. So this is what we saw already yesterday, so therefore this is just a reminder to put it into the context now of what is the response of ecosystems to climate change. We have discussed this at length. So this model yesterday, so which is the paradigm in theoretical ecology, the Rosenzweig-MacArthur model as the predator-prey system in theoretical ecology, where we have used only the region in parameters, so where we don't have any bifurcation, so where we have the coexistence of the two species here. And we have changed here the carrying capacity, not the growth rate. We could have, of course, also taken the growth rate. So we started here off with the carrying capacity, which is the habitat quality, which we change by land use change and climate change as discussed yesterday. And then we end up with this picture, which we had yesterday already, so that from the same initial condition, we go either to tracking, so which is here. So if we have a small rate of change, or we go to tipping so with a high rate of change. The difference here is the response in terms of ecology. Because in the first model, so the outcome was there is an outbreak of a population. Here, in this model, it's the extinction of a population, because the prey gets extinct. It goes to this critical manifold here where the concentrations are extremely low. And this is an extinction. So again, here's our canard, and here the canard is moving. So you see again the same thing. So we are changing the rate, the canard is moving, and a formally tracking trajectory goes to a tipping trajectory. So it's all the same. So as in the first model, which I have explained, and we saw this already yesterday. So this is only just to remind you, so what we have discussed yesterday. So this is now an extinction of a population. But now we would like to go a step further. So because we are looking at living creatures. Living creatures can adapt. This is their big advantage. So compared to so to say, technical things, they can, so to say, deal with climate change in a way. Okay, so, and we asked ourselves whether evolution and adaptation, so fast evolution, is able to prevent tipping. So can we imagine, so to say, that that evolution is really working against the change in biodiversity, against extinction? So this is what we would expect. So I said we have three possibilities, or the species have three possibilities: adapt, move, die. So these are the three options. And so therefore, we looked at the evolution of the system and now change one internal parameter of the system and look so how this would work maybe against it. So this is an approach which comes from evolutionary theory. So this equation, and it has three terms. Basically, there is a genetic variance. There are physiological limits, because parameters which are internal parameters of the species you cannot change at will, so to say. And there is the fitness, because evolution is usually oriented to go to an extremum, so where you have the fittest organism will survive. And this is basically what is in this equation. So it's an evolutionary theory. And this has been done by Cortes and Elner, so also for the Rosenzweig-MacArthur model, but they wanted to see whether oscillations, which are also taken as a threat of a species, because you come very close to the axis where the population density is very low, so that you have also the threat of extinction. So they try to see whether evolution can, so to say, remove these oscillations. And indeed, so to say, you can do that. So they also looked at a third parameter, which is a trait of the species, and looked so whether a trait change would really suppress this oscillation, and indeed this can be done. And this theory of rapid evolution was inspiring us to look at the following thing. So because in our system also, so the trajectory goes to zero. So because we have a population which dies out. And evolutionary theory has shown so that if you include evolution, so your trajectory, which goes, so to say, beyond an empirical population threshold or extinction threshold, can be rescued by evolution. So there is something which is called evolutionary rescue. And this evolutionary rescue is known to biologists already for a very long time, so that evolution can really, and adaptation, which is fast evolution, can really do that. So in principle, this is a stochastic theory, so you would look at whole distributions, but our equation describes only the movement of the optimum, so to say. So our trait our trait is the maximum of the distribution and moves to the optimal trait. So this is what this evolutionary equation which we use is really describing. And therefore, our goal was to prevent this tipping by, so to say, evolution. Here is our extinction threshold. And now we have, of course, deviated because we are solving a biological problem, not a mathematical one. And therefore, we have basically changed the definition of what is tipping in this system. So because we have adapted it, and tipping in mathematics would be when the trajectory goes over the fold or under the fold, so depending on the model system which we had, this would already be tipping. So for mathematicians. But for us, not. So because if we go over the threshold, but we dip only a little bit, but the population would recover. So this is, in an ecological sense, not a tipping because, oh, okay, so the population goes a little bit down, who cares? So as long as it really recovers. So therefore, our tipping is now, so to say, you have to go under the extinction threshold because then the probability is extremely high that this population dies out. And that means it's not crossing the fold, it's crossing the extinction threshold, what counts. And so with this idea, so we have now our population dynamics. We have the environmental dynamics, which is taken explicitly into account, which was not done in classical evolutionary theory. And so we have the trait dynamics, which I showed already with the physiological limits, with the genetic variance, and with the fitness here. Now we have 3 timescales. So we have the ecological timescale separation between the species. We have the timescale, which is the environmental change, so the rate of environmental change, and we have the evolutionary timescale, which is given by the genetic variance. And so tracking would mean so that our trajectory goes to the evolutionary optimum here, along which it tracks, so to say, the quasi-stationary stable state in this system. And what we can see is that we have now compared here the 2 timescales. We have a slow evolution, a fast evolution, slow environmental change, fast environmental change. When both are slow, so then there is a slow risk because everything is tracking. So everything is fine. So the evolution is so to say, working for us. If we have a fast evolution, then we dip even less. That means it's even a lower risk. But now, when we have a fast environmental change, but a slow evolution, then the system will go under the threshold. That means the system tips. But then we can have, so to say, a fast evolution. And then this curve, so to say, this trajectory is lifted. And then we have an evolutionary rescue. So we have a rescue effect on our population. This evolutionary rescue, in our case, is indirect because so the prey is rescued, but due to an evolution of a trait of the predator, in our case. Evolutionary rescue would mean that a trait of the prey is changing, so which is the one dying out, and we have an indirect evolutionary rescue. And when we look now at the timescale diagram, then we see, so to say, that there are different regions. So here is tracking, everything's fine. Then the whole thing would be tipping here if there is no evolution. So, but we see that the evolution, the faster the evolution is, the larger is, so to say, the possibility to really have this rescue effect. And that means a higher genetic variance for the species would be very beneficial for survival, because then these species which have a very high genetic variance are the ones who can adapt best. So this is, of course, what ecologists already know that this is going to happen. This is what you can expect. But so this is now here also coming out in the model. Okay, so as another point I would like to discuss now, also, so has it really relevance? So there are two specific examples, so which I have discussed, very low dimensional. This is not what ecology means. Ecology means we have food webs, very complicated stuff. So we have lots of differential equations describing the different species. And so most of these models are described like this. So you have a species n, xn, so, and you have different gain and loss terms. So these are all balance equations where you have gains and losses. So if you are a plant, then there is a gain term with primary production, so that anorganic nutrients are transferred into biomass. Then there is a mortality term, but then you have grazing, either you are, so to say, the predator, so then you increase your biomass, or you are the prey, and then, so when you are predated by other species, then it's a loss term. And in these type of models, so usually people take different opportunities for these rates. So because you have to formulate the growth rates and the grazing rates in terms of population density. And the two population models I have shown are the most abundant ones in any of these highly sophisticated population dynamical systems, because the grazing rate or the growth rate, so to say, or the grazing rate or assimilation rate, depending on whether you are a plant or an animal. So this would be a Holling type 2. This was the Rosenzweig-MacArthur. This would be a Holling type 3. This was the Truscott-Brindley model. These are the most abundant models in theoretical ecology. So they show up in almost every model. And so also in experiments, you see these type of growth rates mostly in measurements. Okay, so this is quite relevant. These are the two, once again, and they are only, so to say, they only differ in this grazing rate here. So once this is the Holling type 2, this is the Holling type 3, this is Rosenzweig-MacArthur, this is Truscott-Brindley, and both lead to rate-induced transitions. One of them, this one, to a population collapse, this one to an outbreak. But as the good message, so to say, there is fast evolution, and fast evolution might counteract it when the evolution and adaptation is fast enough, so we can prevent such rate-induced transitions. So now, as a third example, I would like to discuss another rate-induced transition, so which is now also related with outbreaks or population collapse, or say diminished populations, but with a completely different mechanism. So far we have discussed the mechanism which is used to discuss the transition when you have only one stable state where predator and prey coexist. Now we go to a system which is really bistable, because rate-induced transitions can also occur in bistable systems. And I would like to show you how the mechanism then works as a rate-induced transition. And we would like to use also, again, a population dynamical system, which is quite common. And this is the spruce buttworm. So this is a creature which is very dangerous for the forests in Europe, and has also outbreaks, has very high population densities, but also very low, and has bistability. Why it has bistability? So because many populations, not only this guy here, but many populations in general, have a so-called Ali effect. So what is an Ali effect? So it means that the population needs a critical density to grow. If the population is below this critical density, then it dies out. But only if I have enough individuals, then it can grow. So this is basically what is now modeled here. And we see that the logistic growth, which is usually used for the population growth, gets another term, and this is the critical density here. So this makes it a cubic function, as we see, and there is a mortality term. So this is, again, a balance equation describing a population growing with an Ali effect. So what you get is a bifurcation diagram, which shows an area which is bistable, and that means we have the coexistence of a low population state and a high population state here. And this would be the parameter. So the parameter diagram, so we have the growth rate here, the carrying capacity here, and this diagram is basically along this black line here. So we are crossing the bistable region, which is in white here. And so this would be the existence of the two stable states alone. This would be the high population and the low population state. So, okay, so now, so far, so good. Now we change again, so to say, a parameter in time. And let me first start with changing, so to say, just to explain the phenomenon, which is important for the whole thing. Let me explain it by changing now this critical density. So we change it again with a certain speed. So this is our rate of change. It's an additional equation. But now we look at the following. So we have a bistable system. And what we have learned already in previous talks is that each of these coexisting states has a basin of attraction. So there are certain initial conditions which go either to this or to that state. And this is a very simple one because this is exactly this type of state which I have shown previously. So there is this unstable state here in the middle, and either it goes here or it goes here. And that means this is the basin boundary. This is location of the basin boundary. It's a very simple thing. And in principle, this basin boundary is nothing else than the unstable solution. So we have two stable solutions. One is the population so the low population density, the high population density, and then there is the unstable one in the middle. Now, so if we don't change any parameter in time, so then I have color-coded here the different initial conditions. So this would be the extinction state, which is stable. The cyan one is the high population state. So this is also not an extinction because we have, so to say, no, it is an extinction, in fact. So when you, no, wait, wrong direction. So that means we are going to the low and to the high population state in red or in blue. Now we are changing our environmental conditions and we are changing the critical concentration here, so for which the population can die. So it's moving in time, and then it's constant, because there would be a bifurcation which we would like to avoid. But now we see what happens here is that many of the blue trajectories can cross now the boundary, because this is the basin boundary. This is nothing else than a moving basin boundary. And the trajectory can cross the boundary. In an autonomous system, this cannot happen. But in a non-autonomous system, it can. And that means what we have is a basin boundary crossing here. And again, with a critical rate, so to say, for each of the initial conditions. So it depends, again, on the initial conditions. And you can either cross at the saddle point or you can cross at the boundary. So for when you start from the stable state, so then it's called a basin instability. So in the work of rhetoric, we don't do that here. So we are not starting We are not starting in one of the stable states, so we are starting arbitrarily. So therefore, it's just a rate-dependent basin boundary crossing, what we have here. So, and now let's look at this basin boundary crossing. Now we have understood what the mechanism is in a bistable system, how you can have a tipping. You go to another state even though you're not reaching the bifurcation. So you are crossing the boundary. Now let's do that, and let's think about a scenario where we have pulse-like perturbations. So you have, for instance, an extreme event, there is a flood, or there is a drought. So all these things have been discussed here already in this conference. And that means, so we are changing now our carrying capacity, and it makes a dip. So here would be the bifurcation. So it would cross the bifurcation. The trajectory would cross the bifurcation. And then, of course, so this would be the bifurcation diagram, basically. So because there is a stable state, there is a saddle node bifurcation, there is the unstable state. And so there is the other stable state is here on this line. Now what happens is, of course, when I go over this saddle node bifurcation, so this is the situation where we would have something like this. So this is then the unstable, and this, and we would go here. So, and the yellow is the trajectory. As I already explained yesterday, so it depends on the rate. So how fast you go here. So usually you have a delay. So you are not just jumping down, but it depends on the relationship of the timescales of the ecological system and the change of the rate. So how it would look like. So here we see our trajectory. It goes to the stable state. And then, so to say, the pulse comes. And now the pulse goes over the threshold. That means the saddle node bifurcation happens, our trajectory goes down and would tip. So depending on what the time scale of this situation is, because it can tip and end up in the low population state. But we have a pulse-like perturbation. That means our conditions, environmental conditions, get better again, because this is just an extreme event, what we have in mind, and it goes back to the original state. So the saddle node bifurcation comes back, but it comes too late. So, because we are already here. So because depending on the rate, so to say, we have already crossed here. Now, when the rate is different, so this would be a slow rate. So because the rate would be the slope here. So this is the rate. So that means it goes slow. And so there is a big time interval between, so to say, it starts and it ends. If we make it faster with a larger rate, so then it goes faster down. It's the same depth as you see, and goes faster up again. So then the difference between the saddle node bifurcation, which makes the high population disappear, is here, but it comes back earlier. So the trajectory has not yet reached because there is a time scale involved here. Again, the ecological time scale, it has not reached yet the low population state. And that means the saddle node bifurcation, which appears again here, makes the trajectory cross the boundary. So because now it crosses the saddle at this point. So the boundary has appeared again, and that means now the trajectory is again in the basin of attraction of this state and goes back to the original state. Now we can understand how tracking happens here. So tracking can happen by a basin boundary crossing, and you go back to the original state. So this would be a bifurcation tipping. So what would come up here? Because we go over the bifurcation and the system tips. In this case, so to say, we have also a bifurcation tipping, but at the same time, At the same time, we have a basin boundary crossing a little later. And so this removes my tipping, and we have instead a tracking. So now let's look at two pulses. So let's look at the situation where we would have, in principle, a tracking. So there is an extreme event, okay, so it hits, and so with a large rate, and we track, basically, so we think the system is safe, fine, oh, everybody is relaxed. There's another pulse coming, a second pulse. It's a very slow rate. It does not even cross the threshold. So it's a very minor disturbance, because you see, so the threshold is here. So this pulse is very slow, and it does not cross the threshold. What does the trajectory do? Again, the time scales come into play. So because now the trajectory goes here, it crosses the boundary, but it's slow in getting, so to say, to the next, in getting back, so to say, to the original steady state. And then the second pulse starts, even though it's not crossing the threshold, it makes the trajectory crossing the boundary again, and it tips. And this is particularly dangerous, because now we have seen, okay, the first pulse was strong, and so we think we are safe because the system recovered. So therefore, we are not paying too much attention to this pulse because it is just a minor disturbance, we could think, but there is history involved. The history is the system was already disturbed once, but it recovered, but this recovery is not fast enough to be, so to say, strong against the second pulse. And that means the system tips, but so to say, but the pulse, which was the one which makes a tip, is much, much smaller, and is not even crossing the bifurcation threshold. And so you can explain it, of course, also in terms of the potential. So you can move these kind of potential wells, and then you can explain it once again, so to say, in this. But so the trajectory, so this is quite obvious what's happening here. So, and this brings me to the end. So non-autonomous dynamics, where you really take into account the explicit time dependence of the change of your parameters. It could be external forcing, or it could be, as in the evolutionary example, it could be an internal parameter of a species or whatever, so which is changed with a certain rate. The time scales are of crucial importance in these things, because everything what I was talking about is a question of time scales. Who is faster? So there is a race between the trajectory and the saddles and the attractors, which are quasi-stationary. Everybody is moving in state space. This is what we have to keep in mind. Everybody is moving in state space according to the environmental change, but everybody is moving with a different speed. And this makes it so complicated to really, so to say, understand the phenomena. And we see new types of bifurcations, rate-induced bifurcations, and basin boundary crossings. Both play a very important role here. And what we learn, so to say, for ecology is, of course, too fast anthropogenic destruction of our nature is something we should never do. There is some hope, because rapid evolution can cure some of the things. So there is a possibility so that a species can adapt, but they have to adapt fast enough. Again, the timescale matters. And basin boundary crossings are something which people are usually not aware of, because this all involves unstable states. Normally, you cannot even observe these unstable states. And this makes it particularly dangerous, because they are difficult to analyze and to keep in mind, but they are also moving in state space. And this is what we have to to keep in mind. And so also the boundaries move. And this is something which brings a totally different new bifurcation into play, these base and boundary crossings, which you cannot get in an autonomous system. And with this, I would like to thank you for your attention. It depends on the species. There are many species known nowadays with a very fast evolutionary timescale. This is also why we try to do an experiment, in fact. So the whole evolutionary paper was inspired by an experiment which we wanted to do together with biologists. So we set up really such a system, and we failed. We could not get rate-induced tipping in this system. And then we discussed with the biologists also why we are not getting this. Because we did everything how it should work out. And then they came up with this idea. So saying, okay, no, these creatures are probably fast enough in adaptation.
Time scale for the prey and the predator need not.
No, they don't need, and they don't need to be the same. So in this example, we have used an evolution for the predator, so we could have also taken an evolution of the prey, and the timescales would be very different. Yes, normally they are not the same.
Condition. So that's also controlled by the environment and the.
The initial condition is something you do yourself, so to say, because you start your experiment with a certain setup, and this is the initial condition. And this can be measured. So, I mean, in a real experiment, you can really control this. Not everywhere. So in many technical systems, I know it is partly very difficult to tune initial conditions and to study basins of attraction, for instance. But in biological systems where you do everything in the petri dish or so you can mix the concentration, and therefore you have really the opportunity to do this by yourself, so to say, to choose the initial condition. Yeah. Yeah. Once again, please.
What happens to the humans when they go extinct, or
what happens to the humans? I hope they don't get extinct. That is what I hope.
not in our lifetime?
Not in our lifetime, yeah. Yeah, so probably not in our lifetime, and hopefully not in the lifetime of our children and grandchildren, but you never know, I would say. So because if we go on with the destruction of the earth as we do now, then so there will be some time when it becomes very difficult to live on this planet. Yeah, so there are more factors, as I said, because species can also move, and this is what they are going to do. And this is what they are going already to do now. So because now when you think about, and this is a slide which Jürgen likes to show in almost every of his talks nowadays, so now we have the threat to humans in terms of floods. Because most of the major cities on the planet are on a coastline. And these are, so to say, threatened by floods. And there will be, and this is what you can already, so to say, expect in the near future. There will be lots of refugees just trying to escape from climate change from those regions which are going to be flooded to other areas. So that means move is also a very important factor, which was not here. So because this was not a spatial system. But in a spatial system, you have these three aspects. So here I have only touched basically the environmental change and adaptation. But species can also move or die. And moving will be a big problem in the near future. This is what I'm afraid of, because most of the population lives in coastal cities. And if the flooding goes on as we see it now, then this is a major threat. I think in Indonesia, they have already moved the capital, if I'm not mistaken. So because of these reasons. Yeah, so this is a real threat. So one has to do something about it. And species will do it. Species move, and this is what we see already. So we see species bioinvasion is nothing else than species moving to another habitat where environmental conditions are such that they have better living conditions and growth conditions. This is why they move. So they move forward north. So to avoid the heat, so many species move northwards. And this is what you can see already now. So that you see certain species which are used to be in the subtropics, they are now in the north of Europe where they are not native species, but they are there.
Back at